Hazal Koptagel. I'm a PhD student at KTH Sweden, and I will present the work I, Oscar, Harald, and Jens are working on. And we recently have class joining our project as well. It is called a fast and accurate method for sampling phylogenetic trees using the neighbor joining algorithm. So we are working with multiple sequence alignment data of N species and M sites as shown on the right side of the screen, and each site consists of a nucleotide ACGT or dash the gaps introduced during the alignment. And there are various nucleotide substitution models such as NJUKE 69, Kimura 80, etc., which are described by stationary distribution pi and the rate matrix Q. And with different constraints on this pi and Q, we have different substitution models. And once we have this alignment data, we can create a pairwise distance matrix with respect to the substitution model of our choice. And then, for instance, we can use neighbor joining to get a tree topology. But this usually gives us a single tree because we have a single set of input data. And alternatively, we can do bootstrapping, where we create k replicates of alignment data by sampling sites with replacement. And then for each replicate, we can compute the distance matrix and then do the neighbor joining algorithm and get a tree. And another work I want to mention is JC sampler that we introduced in our previous project. So what JC sampler does is it samples distances directly from the NJUKE 69 substitution model. If we have M sites and we have two sequences and the number of substitutions between these sequences are phi, or the number of expected number of substitutions. And let's say P is the probability of no substitution, and then the likelihood between these sequences are shown on the right. And we can introduce beta distribution as prior to P, which will give us a posterior distribution, which is a beta distribution due to the conjugacy. So if we have alpha, beta, which are the parameters of prior, and we have m number of sites and phi number of expected substitutions, then we can sample as many p's as possible. But this sampler was sampling distances, not p. Thanks to the NJUKE model, actually p is a function of d, as shown on the lower left side. So if we sample p, we can easily map it to a distance. So we can sample distances between two sequences. So what we do in our project, which is called NJUKE, is we combine JC sampler and neighbor joining. So we have the alignment data again, and then we create a pairwise matrix, which is the expected number of substitutions between each sequence. And then we use this matrix to sample k different distance matrices and do the neighbor joining. Simple as that. But why is it a faster approach? So to get k different distance matrices, our method takes mn square time to get the substitution matrix, and then kn square times to get the k distance matrices. Whereas in bootstrapping, it takes k times m times n square time. And with larger number of samples and longer sequence length, our method is much faster. So the final part is the neighbor joining part, which is exactly the same for both of the methods. But of course, we can make it even faster by using, for instance, FastNJ. So we run our method on a real biological dataset called DS1, which is commonly used in phylogenetic inference literature. And we ran Mr. Bayes for 2 billion generations and discarded some samples as burn-in. And then we used the majority consensus tree, which is shown on the right, as our ground truth topology. And then we compared the transfer bootstrap expectations, like TBE scores of each edge, between NJUKE and bootstrapping. So this score is between 0 and 1, the higher value the better. And it describes how well our samples match the reference topology. And we can see that both methods has similar edge supports. And then we compared our method with, again, our method, but with the higher likelihood trees from our method. And we see that our higher likelihood trees has even better edge support on the ground topology. So to summarize, we introduced this new method called NJUKE. It has a Bayesian foundation thanks to the JC sampler part, and it is a fast and accurate approach, and it provides diverse trees, and it can be used as a standalone analysis. For instance, you can do majority consensus analysis, or it can be used as an input to more advanced phylogenetic inference methods such as Wi-Fi, VBPI, and VPINF. So right now we are working on extension to other substitution models, and we are working on further improvement of our sample trees. These are some references I mentioned throughout the presentation, and thank you for listening.